Okay, well, I think we can make a start. So, good afternoon, everyone, and welcome to the Palaeoverse Lecture Series. This lecture series is focused on software development and methodological approaches in paleontology, with the aim of promoting the use of new tools in the field and to give the researchers a platform to showcase their work. With this series, we hope to shake up the usual type of lecture by having a mixed format, with time dedicated to the presentation, demonstration, and discussion of the software or method presented. Typically, these sessions will run for about 50 minutes in total, and we will run this series throughout the year on the last Thursday of each month. You can already see the great lineup we have for the next few months, and we're really looking forward to all the upcoming talks. If you are interested in presenting a new method or software you have been developing or implementing, please feel free to reach out to us. So before moving on, just a quick few introductions. My name is Lewis Jones, and with me today I have Erin Dillon and William Gertie. The three of us are on the development team of Palaeoverse, an organization aiming to bring the paleobiology community together, to share resources, reach agreed standards, and improve reproducibility in paleobiological research. If you want to know more about Palaeoverse, please check out our GitHub organization and our website, which those links are just going to drop into the chat now. But that's all from me today. Erin will now give you some helpful tips and instructions for this webinar, followed by Will, who will introduce this month's speaker. Thank you very much, and please enjoy the lecture. Over to you, Erin.
Wonderful. Thanks so much, Lewis. So as Lewis mentioned, the format of this seminar will consist of a talk by our speaker, followed by about 10 to 15 minutes at the end that are dedicated to Q&A and discussion. My fellow hosts and I will help field your questions and moderate this discussion. Please note that the seminar will be recorded and that it will be made available on the Palaeoverse YouTube channel. However, we will stop the recording before the Q&A period in case you prefer not to be recorded. So before we get started, I'd like to quickly point out a couple of useful Zoom controls at the bottom of your screen. There should be a Q&A button, which you can use to submit your questions to our speaker. And during the Q&A session, we'll either read out your question or invite you to turn on your microphone if you'd like to ask your question directly. There's also a raise hand button. So alternatively, you can raise your hand during this period and we'll call on you and invite you to turn on your microphone so you can ask your question. There should be a chat function at the bottom of your screen. So currently you can only chat with the hosts in case of technical difficulties, but we'll allow you to chat with other attendees during the Q&A period. And then there's a closed captions button that you can turn on to view the transcript at any time. So lastly, I'd like to highlight a couple of ground rules. Our aim is to cultivate an open, respectful, and inclusive environment during this lecture series where we can learn from each other. So with this in mind, we ask that you please be respectful and kind when engaging with the speaker and fellow attendees, particularly when asking questions and interacting with others in the chat. And any form of harassment or inappropriate behavior will be grounds for removal from the webinar platform. So for more information, please refer to the Palaeoverse Code of Conduct, which can be found on our website. And I believe the link is also going to be put in the chat if it isn't already. So with that, I'll pass it over to Will, who will be introducing today's speaker. Over to you.
Thank you, Lewis and Erin, for introducing the series and all of the things we need to know about today's lecture. I now have the honor of introducing today's speaker, Laura Mulvey. Laura did her bachelor's degree in analytical science at Dublin City University, before moving to Germany to undertake a master's in geosciences at Friedrich Alexander University, or FAU, in Erlangen, just outside of Nuremberg. She's now a PhD student in the Geozentrum Nordbayern group at FAU, supervised by Professor Rachel Warnow. Laura's work revolves around developing Bayesian phylogenetic methods using paleontological data, particularly for complex evolutionary processes. Her previous work includes modeling coevolution between parasites and their hosts, and drawing evolutionary signal from morphological characters for phylogenetic inference. Today, we are privileged to hear Laura present a talk titled Assessing the Adequacy of Morphological Models Used in Paleobiology. Please join me in welcoming Laura to our virtual Palaeoverse stage.
Thank you for that introduction, Will, and yeah, thanks for the whole Palaeoverse team for organizing these lectures and inviting me to contribute one. So yeah, so as they said, I spent a lot of my PhD working on like phylogenetic methods and thinking about how we integrate data from the fossil record into Bayesian phylogenetic analysis. Yeah, so basically, we see phylogenetic trees throughout the literature, quite a lot, and they're quite useful for understanding how species are related to one another, like organizing our thoughts and like description about how, species has evolved through time. But they also give us, basically an evolutionary hypothesis of like the evolution of a group, and they allow us to start to make inferences about the relationships of the different species, the rates of evolution, different patterns of evolution through time. Sorry, I'm just going to move the cat. Yeah. Yeah, so basically, when we are trying to construct an evolutionary tree, we need some character data. And a lot of phylogenetic methods have been developed and kind of focus on molecular data. So they might involve, describing how molecular character information has how it evolves. As paleontologists, basically, when we're working with different groups, we don't have molecular data, right? So we don't basically find fossils ready to be, with their character data available and ready to be sequenced. So we have to use morphological data and try to understand morphological evolution. And I mean, morphological data was, the first type of evidence that was used in phylogenetic analyses. But since, a lot of more like Bayesian type analyses focus on molecular data initially. So yeah, then we always need to think about how do we actually model morphological evolution. And there's different like model components available in morphological or in Bayesian analyses. So basically you kind of have this like tripartite approach quite often, especially if you're doing some like dating analysis. And these different components basically describe, essentially like different aspects of, the evolution of your character data. So, if you have your clock model, that basically describes how the different traits have evolved, like the time, like the different rate of change. Your tree model basically kind of tells you what was done. So what can happen to a species during its time. So there can be species origination, species can go extinct, and they can be sampled through time. And this would be, if you're doing, say, like a fossilized birth death analysis, this is where that part comes in. So that's like the tree model part. And then you have your substitution model, and that basically tells you like, how characters change. So how characters in your alignment kind of like change and evolve through time. And this is the part that I focused on a lot during my PhD so far. And that, I'll be talking about mainly today, or well, entirely today. So basically if we look at if we look at morphological data that we use for these types of analyses, this is an alignment, and it basically describes, it's from Cambrian echinoderms from, Cambrian stalked echinoderms. And essentially down here we have, can everyone see okay, so basically down the side we have like the taxa, and across here we have the different character states, different character traits. And this is basically all the information we have to say, to try to make some fundamental inferences about the evolution of the echinoderm body plan. So like we really need to make sure that the methods that we're using are fitting to our data. Because we don't always have a lot, and it's important to really maximize what information we can kind of get out of these alignments. But if you kind of look at the data itself, it's quite complicated. So you have, so for example, this trait describes the presence and absence of some kind of morphological characteristic, right? So 0, we don't see the trait in this organism, and 1, we do. However, the 0s and 1s here kind of refer to different appendage branching patterns, so different morphological feature. Here we have different plate arrangements. So you can kind of see already, like within the same even the same alignment, we're getting quite different biological meanings for a 0 and a 1. So trying to come up with a model that basically explains those differences is it's not very straightforward. And also we have all these like kind of special characters in our morphological matrices. So we have basically lines like these, and these denote characters that are non-applicable. So essentially that would mean that, you know, so one of the other character traits basically says the presence or absence of a certain trait. And then, you know, another trait might go on to describe that trait in more detail, something about the ornamentation of it, for example. Whereas if, you know, a certain taxa doesn't have that trait at all, then when you go to describe it, it's non-applicable. And then we also have a lot of missing data in morphological matrices. But the current kind of substitution models don't, you know, always take these into account. And I mean, there's new models coming around, like embedded dependency type models that do, but yeah, quite often we don't really take into account or know how to deal with the missing data or the non-applicable data. So yes, basically I wanted to kind of talk to you today about different substitution models that are available and like how these kind of impact different parameter estimates that we'd be interested in. And then kind of approaches for figuring out, okay, so how do we know if like any models are actually fitting our data or if they make sense? And then yeah, to go through a tutorial on model adequacy. And yeah, everything that I'll be talking about today basically comes from this preprint that's out. There's a QR code for if you're interested. But yeah, so this is basically the first chapter of my PhD, and it's out in a preprint now, but it's in revisions now, so hopefully it will be out quite soon. Yeah, so basically, as I said, the substitution model describes your character change in your alignment, right? So if you're trying to, you know, infer some tree, you need to basically tell the model how you expect the characters between different taxa to change. And as I showed you, the morphological alignments are filled with kind of all these different special cases. So it makes it quite difficult to generalize how you would expect things to evolve. So due to this, we mainly rely on the MK model, which if anyone knows any like molecular substitution models, this is like a generalization of the JC model. And it makes like rather simplistic assumptions about the transition of characters in our data set. So basically says, yeah, okay, you can transition from a 0 to a 1 or a 1 to a 0 with the same probability. And also that, you know, so sometimes you have binary characters that are just 0 and 1, but you can also have multi-state characters. So like more, yeah, more complex numbers of characters that also, yeah, will also have the same probability of transitioning from one to the other. But, you know, you might want to make these, yeah, kind of relax some of these more harsh or more kind of strict assumptions. So people often use like add a gamma rate variation, and this essentially allows you to say that like, you know, certain traits might transition at a different rate. So, you know, like you might have some more plastic type traits that are evolving faster relative to others. So then, you know, having a gamma rate variation allows different traits to basically transition, you know, at a different rate. It's also common to basically partition the data. What that means, basically, you'd be like cutting up your morphological matrix into, you know, different groups, and then you essentially apply an MK model to each of these different groups to kind of say that, okay, I think all these will kind of have a similar, you know, evolutionary rate, and, you know, all these different traits. And I mean, the most common one that people use is basically partitioning based on the maximum state. So, you know, you put all the binary characters into one and give them one MK model, and then all the tertiary into another, and so on and so forth. People also might want to, you know, partition based on like anatomical information or region or, yeah, things like that. And then you can also make this correction for ascertainment bias. And what this basically does is, you know, like when you're coding a morphological matrix, obviously it's like a lot of work. So you want to pick characters that are informative. So they basically, you know, point out like differences between the species or between your different taxa, because, you know, you're not going to just code for characters that are, you know, across the whole data set, because, you know, where do you stop then? You know, it's like you basically describe the entire species, and it just, you know, it increases like the workload and doesn't really add any information. But this is quite different to, you know, a molecular sequence. So if you have, you know, if you sequence some molecular data, a lot of your sites are going to be like they're not going to be any different from each other across all species. You're going to have conserved sites. So then when we take, different substitution models that were made with kind of molecular data in mind and use them for morphological data, it looks like, given that every site has like one kind of character change, it looks like there's been an awful lot of evolution kind of happen between your different taxa, and you end up with these crazy long branch lengths. So this correction came about to basically condition on the fact that all of your taxa are varying and kind of avoid this telling the model that like we're expecting all the sites to be different and that it's that it's okay. So this is also, of course, not like an exhaustive list of all the ways you can kind of relax the substitution the substitution model or all the different models. But in the study, this is kind of like what we focused on, and we wanted to know like, okay, so these are the different extensions that we can have. Like, do they make a difference, and how do they actually like impact the parameter, the different parameter results that we get? So to do that, we basically took I think 120 odd tetrapod data sets, and then we just did an inference to under like different combinations of these three different extensions, and tried to see whether or not they were making like a difference to our parameter estimates. So this plot here basically just shows you the tree length. So basically we took like an MK model, so ran it with the most simplistic model. There's no assumptions relaxed or anything like that. And then we did same we took all the same data sets and ran it with an MK model that allowed for, gamma, then ascertainment bias, gamma and the ascertainment bias partitioned based on the max character, max observed character, and then variable, and so on. And basically we just wanted to know like, okay, so are these different choices in substitution model impacting our tree length? So this plot here basically shows any of the plots that are on, or any of the plots that are on the right-hand side show that they're producing trees that are longer than the MK model, than like that for the MK model. Whereas if they're on the right, on the right bigger and on the left smaller. And this is like the percentage difference. So you can see like some trees are like, like quite substantially like bigger depending on like what model you're using. So this is our first time that like, like these models are really having quite an impact. But this is basically a summary. On the left-hand side, it's like a summary of, over the whole 120 odd data sets. And we want to look at them individually and say like, okay, so are these patterns consistent? So these two panels here at B show the differences in tree length for the different models for two different just individual data sets. And then we can see like, so while the histograms might look like there's some kind of general pattern kind of emerging, if you look at the data sets individually, like we do see kind of different responses to the different morphological models. Whereas like here it looks like the partition models are always giving, bigger trees. Whereas here, like for this data set, we see like the MK model is actually larger than the partition models. So even though we're seeing differences, they're also not like systematic differences. It's not like a consistent pattern. And then these plots down here basically show the Robinson's fold distance. And they show basically like the tree space. So the models are also impacting our topology. So basically, like understanding like the tree length, like how much like the evolutionary distance between species and the actual like topology that we're inferring are both being impacted by by our choice and substitution model. So it's quite important to think about, okay, how do we actually choose a model? So when you're trying to choose a model, you have your alignment, you need to do something, you want to get your tree. And quite a common approach, or what people often think of, is to use model selection. And model selection in phylogenetics usually involves, calculating your marginal likelihoods, or estimating your marginal likelihoods, and then calculating your Bayes factors to choose between 2, 3, 4 different models. But what's important to kind of think about is that this gives you like the relative fit, right? So it tells you out of, the 10 models that you're testing, this one is the best model, but doesn't really give you any intuition of like whether any of the models actually fit. So like by doing this, you're already making an assumption that like the model, like at least one of our models fit the data. Where in reality, it could be that like none of the models fit very well, but the model that you pick happens to pick, or happens to fit the data slightly less poorly, but still isn't a very good fit at all. And using model selection approach, we'd have no idea of that. There are also some other issues with model selection that I'll get into quickly at the end, but yeah, basically we wanted to take a model adequacy approach to kind of figure out, okay, what's the absolute fit of a model to our data set. And we also thought this was quite important for morphological data because, all the different alignments, like they're quite bespoke, right? They're made by, a couple individuals, and yeah, kind of thinking that, some of these more are quite simplistic models will always, that one model will always fit all these different data sets. We weren't really sure. So we wanted to know, like, okay, so are any of the models actually doing a good job at fitting our data at all? Yeah, so the kind of model adequacy was the most natural kind of way to test this. And we worked with the model adequacy approach, was posterior predictive simulations. And I did this in RevBase, described in this paper by Sebastian from 2017. And basically posterior predictive simulations, it works by you have your empirical data set, and then you do like a normal MCMC inference to do like infer your trees. And while you're doing this like Bayesian inference, you're sampling from your posterior, so you're kind of pulling values that will be important. So things like branch lengths, trees, if you have gamma, like different alpha rates, so like things like that you want to sample while you're doing your MCMC. And then you can use that information that you sampled to simulate new data sets. So you'll simulate, say if you did your first analysis under an MK model, then you'll simulate data sets under an MK model using like the values that you got from your posterior as input. And then you can go on and basically do inference of your new simulated data under the same model. And then you can compare the results. So like the kind of like overarching idea here is basically if your model is doing a good job at like capturing the underlying dynamics that like kind of gave rise to your data, when you simulate under the model, you should be getting quite similar, similar results. So basically, if you compare your simulated data to your empirical data, like they should be quite similar. Similarly, like if you compare your inferred trees to like your empirical or so trees, you think they should be quite similar if the model is doing a good job, fitting your data. But yes, basically by like similar, how do we actually quantify similar? So with this we use like test statistics, and they basically it's a way of like numerically summarizing your data. So like how you actually take a morphological matrix or something, get one number out of it and then compare it with your empirical to get one number out of it and see how close these two numbers are. So for posterior predictive simulations, we looked at data-based, inference-based, and mixed. And I'm not going to go into the whole simulation study for this because it's not important for actually using, well, and it's not that as, basically we just did like a load of simulations where we simulated under a model. And then we used different test statistics to try to see if we could fit which ones were like indicative to which models we simulated under. So for database, we used disparity metrics. So we looked at like Garrer's coefficient and generalized Euclidean distance. For inference-based, we used tree length and tree inference. And for mixed, we used consistency index and retention index. Ultimately, we found that the mixed test statistics were the only ones that really we were able to figure out which model we simulated under. So really we're kind of capturing these dynamics. For the database and inference-based, basically we weren't ever able to determine which model was correct. So so we went forward and said, okay, we can use these mixed test statistics because while this whole pipeline is very nice, basically, unless you can basically have a test statistics that compares your two, compares your, your empirical from your simulated data, you still can't really tell anything. So yeah, we found that mixed test statistics were the best for that. And as I said, I used the consistency index and retention index, which consistency index measures the amount of homoplasy or like convergent evolution, and the retention index measures the amount of synapomorphies. And to do this, basically, we took our empirical data set and just our MCC tree, so our summary tree, and you can use both of them to calculate your consistency index and your retention index. And then for all of our simulated data sets, we did the same, and we used the empirical MCC tree, which also meant we could skip the last inferences, so we didn't have to do inference under on all of our simulated data, which also speeds up the whole thing like quite a lot. Because yeah, if you're doing like if you want to do a model test, then you have to basically do your first inference and then simulate your data, and then do your inference under like 500 of your simulated data sets. It can just cause your analysis to go from like, I don't know, 10 minutes to like a couple of days. So yeah, it's a lot better now that we don't have to do that. So yeah, so maybe it's a bit difficult to kind of grasp, but basically when you have like so you do you simulate your data, and then you calculate your test statistic for each of your simulated data sets. So for example, you have your one empirical data set, and then you could have 500 simulated data sets. And for each data set, you want to calculate, so this is a histogram here of our consistency index. So we ultimately have like 500 simulated consistency index and one empirical value. So this green number here is the consistency index calculated for our empirical data set. And then this histogram shows our simulated set, and then this blue line here is like the median value for our simulated data. And ultimately, we want those values to be as close as possible, because the more similar our simulated data is, the better the model. So yeah, basically, if you were to do like the tutorial that I'll show you at the end, basically you have your two test statistics that we validated, and you want to say like, okay, does this model, so like, does the MK model fit my data set? So you do your inference, and then basically you can validate them using effect sizes or p-values. I kind of like using effect sizes, because I mean, if you want to like compare different models, and I just find them like slightly more intuitive. And I mean, especially for when we were doing the study, it allowed us to compare the performance of different models. But if you're just looking at one model, using a p-value is totally fine. And here basically, you calculate your effect sizes, which is your empirical consistency index value, or your empirical test statistic minus your simulated test statistic over the standard deviation of all your simulated values. And essentially you just want it to be close to zero. So the closer to zero, the model is adequate. And we kind of determined that essentially if the minimum and maximum of these box plots here kind of cross that zero line, then you can consider the model to be adequate. And then obviously for a p-value, if your value is greater than 0.025 and less than 0.975, it's yes, it's adequate, because basically they're not significantly different, the two populations. So that's kind of ultimately the idea of posterior predictive simulations, that you just want to be able to simulate under the model and get quite a similar result to basically similar output than your input essentially. So I just wanted to kind of briefly talk about model selection, because I know that's like the most kind of like standard approach, and like you read people using it a lot to kind of choose between different models, and it's been tested a lot in molecular studies, but I guess it's been tested like somewhat less so for morphological. And essentially, basically, we found that when we simulated data, we were always getting like partition models as the best model, even if that wasn't what we simulated under. We found this very odd. So I'm running a bit long. But then basically it kind of started to make a bit more sense when we looked at how we partitioned data. Because when you partition data in a morphological matrix, it's really, for morphological data, it's quite different than how you do so for molecular. So if you have an alignment like this, and in an unpartitioned matrix, everything is in the Q matrix size 4, right? So then when you start to split it up, you're actually changing the size of the Q matrix. Whereas for like molecular data, you're never changing the size of your Q matrix. You're always going to have 4 nucleotides. But this is entirely different for molecular morphological data. So for this type of analysis, then basically you start partitioning, and then you end up with these like 3 different partitions. And yeah, essentially, you're having different Q matrices of different sizes, which is like a fundamentally different kind of process than you would have for molecular. And this is just to kind of show you that basically, if you're using model selection, you're relying on calculating your marginal likelihoods, but your marginal likelihoods are like directly impacted by the your number of base pairs, so like your number of transitions, or like your states essentially. So when we change the number of states in morphological data, we're having like a direct impact on the likelihood calculation. And this is why basically we can't really use it very well to decide between partitions, because we're always going to get a higher likelihood if we start to partition it up, but it's not actually a biological signal. It's directly from the likelihood calculation. So we can't really make too many kind of decisions. It's not a biological pattern. So say if you were trying to use it to distinguish between different partitioning schemes, it wouldn't really fit as well. And just kind of to show this, we took a data set that has 6 states, and then we basically increase the number of partitions. So on the left here, we have like the marginal likelihood, and here we have the number of partitions. And this one here has just 1 partition. So basically, we have all of the character states just in 1 Q matrix. And then in the second one, we took all the binary characters, gave them 1, and everything else was in a second matrix, and kind of so on and so forth. And we basically divided everything up. And we can see that the marginal likelihood is just increasing as we add different partitions. And if we simulate data in unpartitioned, basically we consistently see this pattern that is driven by the size, changing in the size of Q matrices. And I mean, it's also not like, it's not a biological signal, but we can, sometimes it could be, sometimes it could make more sense. But using, marginal likelihoods to distinguish that, I won't really give you a very good answer. So basically, I guess like from this, I want to say that when we're doing these morphological analyses, like we do really need to think about the model choice, because I guess sometimes like these especially when the matrices can be quite small, and then people just kind of like or it's like kind of the endpoint of analysis when they just want to like get a tree. But yeah, they're really having quite an impact on our parameter estimates. So we need to be a bit conscious of that. And using model adequacy can kind of help us really to figure out if a model is describing our data and what that looks like. And I didn't show any like empirical results here, but we also found that the models are like whatever model is adequate is like, it's quite data set dependent. Like there's not really one model that like fits all. It really depends on like the data. And we're also been trying to figure out like, okay, so are there any kind of key characteristics of data sets that you can kind of figure out which model you might end up using? But like it's really based on model adequacy, it's really hard to say like when a model is going to get chosen. So that means you always need to test. Like you won't just know what model to use. And also we found that there's not always a model that's going to fit perfectly. But that's also, I think, like not surprising really. Like if there was always a model available for all these different morphological data sets, I think that would be almost more surprising, I guess. So yeah, it's definitely important to kind of consider which what type of model you're using. And I can walk you through kind of carrying out one of these model adequacy analyses basically. So there's this is just like the QR code to the RevBase. So you can also do model adequacy in like any phylogenetic software once you can, or any software really once you can simulate under the model. But I'm just going to talk about it in RevBase because that's the software that I use the most. So I'm just going to go to, sorry, I should have this like, Can everyone still see my screen?
Yep, looks good.
Okay, great, thank you. So basically this is like the RevBase website, and there's loads of tutorials here for running different analyses and how to kind of get started to work with the software. But this is the one that I made for doing this model adequacy testing, specifically with morphological models. And basically there's like just kind of explains the different steps of how to go through the analyses. But essentially we can just, yeah, you can just like download these files here, and it will give you everything you need to run an analysis. So if I, yeah, so basically when you download that folder, you'll end up with basically a data and a scripts directory. And your data is just like two morphological matrices that you can kind of play around with. And your scripts are, you'll have like two starting scripts. So this is to run the analysis with an MKVP, like an MK model that accounts for ascertainment bias, is partitioned and allows for gamma. And then the other is just an MK model. You can like switch these out and play around with it what you want, but basically these are kind of just the two scripts to you can use to get you started. So yeah, basically if you go to like the first, like the analysis one here, basically this just kind of reads in your data, the name of your data, so where it is, and then it's always good to kind of give your analysis like a name, because unless you like change directory, or remember, like RevBase will just like write over your files if you don't change the name. And then you might want to change which model you're working with. And then it's very important to like say the number of states that you have in your alignment. So in this one, I know that there's, yeah, you have 0, 1, 2s, and 3s, which is why it's set to 4. And then we basically just tell it to read in this script here. This model script, and what that does, yeah, basically, so this is how like the RevBase kind of language works here. Basically, you want to give yourself some like helper variables. So you'll have like your number of taxa, which will be important, then your number of branches, and then you want to set up your tree model. So in all these analyses that I've ran, they're all like unconstrained analysis. So you don't have any like, yeah, we won't have a clock model, we just have this tree model with a uniform topology. And then you set some moves on it. And then this is basically where we define our substitution model. And for an MK model, this is all the information that we need. We just say we want a Q matrix. And as I said, it's like a generalization of the juice counter model, and then the number of states. So basically a JC model with 4 states. And then we put this into our sequence evolution model. We say basically our tree, our taxa names, our Q matrix, and then we have it standard because we have morphological data. And that's like the main points from the first script. And yes, that should run so if you just type in RevBase, and then if I run this, that should hopefully all run fine. So now basically in this step, we're doing the first MCMC. And while we're doing this, we'll basically be like sampling from the posterior, we'll be getting our tree, and we'll be, we're getting our empirical tree and getting our MCC tree. Oh, sorry. So sure, well, you can see this, but yeah, so then there should be like this output of the MK model that we just tested, and then you end up with different log files, because we have 2 separate runs just to help with making sure that everything's converged. And then we have our .tree file, which will be like our posterior distribution of trees, our log file, and this .var file is basically, it's quite specific to this analysis, and that's the sample that we took from the posterior that we used to simulate, and then our MCC tree. And then in the next part, we basically want to do our simulations. We want to simulate our data sets. And this file here, the main thing that you might be interested in like changing might be this parameter here. And this tells you how many data sets you want. So based on how in your .var file, you have like a number of trees based on how much you basically sampled from your posterior. And this thinning file changes how much you simulate. So now I'm telling it to simulate on every 4th tree, which would give us 500 simulated data sets. And yeah, in the study that I was talking about earlier, basically we kind of found that having 500 data sets was sensitive enough to basically test the give us a good result on which model we needed. So then if we run that, yep, and then we get this file, which basically has, sorry, I can't make it any bigger, which basically has 500 and probably like 501. Yeah. And then we'll have, a data set that has been simulated for this model. And then the last thing you want to do is basically analyze this in R. So sorry, I should have had R open. So yeah, so basically, once you're in the right place. Then you just basically say, which model you're using. You will make sure the name of your data set is correct, and then your analysis name. And then, yeah, basically you want to read in your simulations. So then you read in your simulations. And here we have like 501 that we ran, and then just make a container for your results. And then this part here is to calculate our consistency index and retention index. And this is done using the Cladis R package. Also, we need the ape R package to basically read in our trees. And it's done using the Fangorn package. And yeah, basically, if you just run through this, that will calculate your consistency index and retention index for this model. And then you can basically plot your results if you want to take a look. And then, yeah, from here we can see that essentially, this line here isn't crossed, and we can see these are quite far. So essentially what we're saying is the MK model is not adequate for this data set. And then you can get like a CSV of your p-values as well for your consistency index and your retention index. So then you can say, depending on which ones you want to use, effect sizes or p-values, you can determine, they will agree with each other, but just whichever one you want to use, you can determine whether or not your model is adequate for your one data set. And that's all you have to do really. I mean, it's quite fast, I think, especially since you don't have to do all the other inferences. And then going forward, you're not like comparing models, but you know for sure whether or not, for this given data set, can you use this model? And I mean, the tutorial is on RevBase and all the code is there and should work, but if anyone tries it out and it doesn't work, please let me know. But that was mainly what I wanted to talk about today.
Thank you so much, Laura. That was great. And we really appreciate, you walking us through the tutorial here afterwards. Now we're going to end the recording.